Hi, my name is Darsh Mandera, and I'm a rising senior at Jesuit High School in Portland, Oregon. I've been passionate about research in bioinformatics for the past several years, and I've focused on the field of precision medicine, where we aim to improve patient treatment by focusing on patient-specific information. Particularly, my work is centered on targeted cancer drug prediction, and I'm excited to share my project with you, OncoRx, an integrative approach to identification of pan-cancer molecular biomarkers and prediction of targeted multi-drug cancer therapeutics. The current approach to treating cancer is a one-size-fits-all approach that's ineffective 75% of the time. With microRNA having been identified as a key biomarker of cancer, researchers have been using machine learning to predict targeted cancer drugs based on microRNA. But current research doesn't use real patient data, and it ignores drug combinations, focusing on monotherapy instead. My goal was to create a machine learning program that predicts the right drug combination to give to a cancer patient based on their personal microRNA alterations. Before I dive into my research procedure, I'd like to quickly share a little bit of background information regarding microRNAs and their role in cancer. MicroRNAs are a type of RNA that regulate the process of gene expression. When the microRNA count in a patient is altered, whether it's increased or decreased, the process of gene expression can become dysregulated. This leads to cells going uncontrollably, which we know as cancer. Since microRNAs play a key role in driving cancer, we can use a patient's personal microRNA alterations to predict which drug combination would be the most effective treatment for them. My overall procedure was implemented in Python and its libraries, and it had 4 main phases. First was pan-cancer data preparation, where I extracted and wrangled the exact data I needed to make drug predictions. This was followed by identification of key cancer-causing microRNAs, where I used feature selection to identify the most important microRNAs in the dataset. Feature selection was coupled with functional enrichment analysis, which allowed me to validate that the microRNAs identified in step 2 were truly driving cancer from a biological perspective. And the final step was the machine learning pipeline itself, involving training, cross-validation, and testing to build an optimal predictive model. The result of these 4 steps is a tool that identifies cancer-driving microRNAs in a pan-cancer dataset and uses these to accurately predict targeted therapeutic combinations for cancer patients. I'll go into further detail on each of these steps, starting with data preparation. I used data for 23 cancer types from the Cancer Genome Atlas, which contained over 6,000 patients, 705 microRNAs, and 230 drugs. From this large dataset, I used pandas to extract the exact information I needed, which was how much each patient had altered in their microRNA count in comparison to a healthy human, as well as the drug combination that worked the best on them. The next step was feature selection, which allowed me to identify the microRNAs that played the biggest role in the final drug combination selection. While I experimented with principal component analysis, the extra trees classifier proved to be a much more accurate algorithm when it came to identifying key microRNAs. Feature selection allowed me to go from having 705 microRNAs to 84 key microRNAs that played a fundamental role in the final drug combination selection. In order to validate that the computationally selected key microRNAs were biologically significant, I undertook functional enrichment analysis, which demonstrated the role of the selected microRNAs in critical cancer pathways such as MAP kinase activity, as well as in patient survival over time, validating that the selected microRNAs were truly driving cancer and that treating patients based on these microRNAs would have an impact on their survival. Once the key microRNAs were validated, I built a machine learning classifier, which predicts the best drug combination to give to a cancer patient based on their microRNA alterations, with extra weight given to alterations in the driver microRNAs. I built models using the K-nearest-neighbors algorithm, the AdaBoost classifier, and the 1 versus rest classifier. And the 1 versus rest classifier was shown to have the highest prediction accuracy of the 3 at 63%. To further increase the prediction accuracy, I employed cross-validation, which allowed for iterative training of the machine learning model and selected the best hyperparameters to use for the 1 versus rest classifier. Cross-validation was coupled with statistical techniques like threshold variance and decimal rounding, which eliminated high variance and data homogeneity. All in all, this improved the prediction accuracy to around 85%. Finally, I built and implemented a median scoring method, which calculated the median value of 3 different accuracy metrics as opposed to using just one. This reduced the impact of outliers when selecting the best hyperparameters during cross-validation, increasing the final prediction accuracy to 92%. Overall, OncoRx is an improvement over current monotherapy research, but more importantly, it's an improvement over the one-size-fits-all approach used in clinical settings, since we go from a 25% to a 92% treatment success rate. I'd like to further pursue this research in 3 dimensions. 1. Instead of just using microRNA data for drug prediction, I'd like to use proteomics data as well, which could help improve the prediction accuracy. 2. I'd like to explore using neuromorphic computing as opposed to supervised machine learning in order to improve the prediction accuracy. And 3. I'd like to publish this research in a peer-reviewed journal so that OncoRx can be available to the scientific community. Thank you.